Ayo, is this thing on? All right, it's going. It's the energy lecture. Okay, we're talking about how we're making energy. We've got plants, we got animals, and we have some bacteria in there. Okay, so typically we're going to talk about the mitochondria doing cell respiration. Okay, through a three-step process: glycolysis, Krebs cycle, and the electron transport chain. Okay, so our bigger diagram here, zoomed out, we have glycolysis, the first step, which actually happens outside of the mitochondria. Okay, this is just in the cytosol, the cytoplasm. Now bacteria will do this, and that's all they do to make their ATP. Makes a little bit of ATP, 2 net ATP. Okay, 4 total, but you have to use 2 to initiate the process of breaking apart glucose to give you pyruvate. You gotta break a bond to make a bond. Okay, and so we break apart that bond, and then we get 2 ATP out of it, and we also charge up 2 NADH from that process. Now bacteria will do that, and that's how they make their energy, and it stops there. However, animals are going to do that process, and then with that pyruvate, those 2 pyruvates, the 3 carbon molecules, we have an additional factory called the mitochondria, which is going to do cell respiration and squeeze some more energy out of the bonds in there. Okay, let's make sure that we know what the bonds are. We have, or what the reactions are. So our reaction, first we break a bond so that we can make another bond. Okay, so here's our breaking. This is exergonic, this is endergonic, and this is to make a bond, because energy here on the y-axis is going down for breaking a bond and going up for making a bond. Okay, and so we break the bond in glucose so we can make the bond in ATP or NADH in our glucose or in our glycolysis process, which is happening in bacteria as well as your plants and animals. Actually, all eukaryotes, all prokaryotes are going to go through that first process. Okay, now the ATP is just used by the cell, and then the NADH is going to enter the mitochondria for these other processes. Okay, but most importantly, what's entering the mitochondria is the pyruvate, 3 carbon molecule, which we can break some more bonds and get some more energy out of. So it's going to occur in the Krebs cycle. Krebs is occurring in the matrix of the mitochondria, and then that Krebs cycle is going to charge up 3 batteries. Krebs is pretty cool. Charge up 3 batteries. We got ATP, which the cell can use, and then NADH, which is going to be used in the electron transport chain, and FADH2, which is going to be used in the electron transport chain. Now those are really good things to use for the electron transport chain, which we see up here, because that NADH and that FADH2, they have H's, and we want H's around to concentrate so that it can flow through ATP synthase. If hydrogen is concentrated in one area, which we see here, it's concentrated in the intermembrane space, which is this space here between the 2 membranes. And if that hydrogen is concentrated there, then it can flow through the ATP synthase, spinning it, and that motion is the energy needed to make the bond of ATP. ATP synthase can make that, okay? Now, the only way you're going to get the hydrogens concentrated up there in that space is if you are breaking these charged batteries here from Krebs and then using the energy from the broken bonds. Again, we break the bond and then we release energy and then we have some sort of motion. Now the motion that's occurring is the hydrogens being pumped by protein 1, 3, and 4. Okay, protein 1 is going to break NADH into NAD+ and pump hydrogen. Protein 2 is going to break FADH2, and then protein 3 is going to use the energy to pump hydrogen. Protein 4 is also going to use the energy to pump hydrogen, okay? And then protein 4 is also why we were breathing. We breathe 4-4, and what that means is that your oxygen, the reason you breathe oxygen and send it out to the bloodstream and all of your cells is so it can go to the mitochondria, specifically to the cristae, where the electron transport chain is going to take that oxygen on protein number 4 and pair that oxygen with some extra hydrogens, which of course we have plenty of extra hydrogens around, and make water. So interestingly, water is a waste product of cell respiration when we are trying to make ATP, because that's the whole goal for this thing, making ATP, okay? with ATP synthase, the enzyme, in the cristae, inner membrane of the mitochondria, okay? Now, protein number 4 is just taking the extra hydrogens and the electrons that were bouncing around in there, because we broke these bonds, we caused some movement, and then we're going to form the bonds between oxygen and hydrogen to make wastewater. So you will see that water comes out of the mitochondria as a waste product. That's because of what's happening right here. Now, why do I call this the electron transport chain? And here it's called oxidative phosphorylation. Okay, well, we can talk about oxidation later, but phosphorylation is adding a phosphate. So how do you go from ADP in order to up to ATP? How do you go from ADP up to ATP? You add on a third phosphate. So that's why this is called oxidative phosphorylation, which is probably pretty hard to read, but you know, if you're looking up diagrams of mitochondria and cell respiration, you will see that. Okay? All right, now, I kind of skipped past the Krebs cycle. That's because it's really complicated, and there's a whole bunch of different things, rearranging of molecules in here done by all these blue, purple things. Those are different enzymes which are doing the work, and then these molecules are being rearranged. So we are going to be breaking molecules, and then carbon is going to be kicked off. So this is the simplified, this is the detailed. And so essentially, we're bringing pyruvate in from breaking glucose. We've got pyruvate coming in, and then we're creating acetyl-CoA, and then we have this CoA added, CoA taken apart, CO2 getting kicked off because we're breaking carbons off of the pyruvate, and then we're using that energy from breaking the bonds and rearranging the carbon bonds. We're using that energy to make the bond in NADH, to go from NAD plus to NADH, to go from FAD to FADH2, adding on 2 hydrogens, a higher energy endergonic reaction, which is done by enzymes, okay? It's not magic. There's an enzyme there. You can look into the detail. But in general, we know we're getting 3 different batteries out of there in that second step of the Krebs cycle. And that second step of the Krebs cycle there, the reason we charge up those batteries is so that we can use those batteries up here in the electron transport chain. That's why it's a 3-step process. Step 1, step 2, step 3. This is step 2, Krebs, citric acid cycle. This is step 3, the electron transport chain in the cristae. Okay? All eukaryotes are going to do this. Okay? Now, why do prokaryotes not do this? Prokaryotes do not have any membrane-bound organelles. They're very basic. They don't have a mitochondria. Okay? But one day, a billion years ago, there were 2 prokaryotes, 2 bacteria working together. One swallowed the other one, and instead of killing it, it said, hey, will you work for me? I'll protect you. And then that's what became the mitochondria. Okay? And then the big cell that swallowed the smaller cell, the big cell became the basis of all plants and animals and all eukaryotic organisms on the planet. And that smaller bacteria that was eaten became the basis for all mitochondria. That's the endosymbiotic theory of evolution, which, of course, has been proven. That's just how it works. Okay? All right. So I think that covers that one. I guess I could also just tell you that when bacteria, or when you exercise really, really hard and get super exhausted and your muscles get sore, when bacteria, you need oxygen. It's oxygen we need to talk about, right? Okay, so we see where the CO2 comes out of the Krebs cycle. That's why we exhale CO2, but we breathe oxygen, and oxygen goes in right here to protein number 4 for the electron transport chain. So we know where that comes from. Okay, but why, what happens when we are exercising really hard and we get super exhausted and we run low on ATP? Then we go into the anaerobic threshold, right? And here we are going through a fermentation process, and the whole point is to regenerate NAD, because we're using the ATP, but we don't use NADH that often, okay? Or we certainly don't use it that fast. So this is a much better option to use the mitochondria, to take in oxygen, to make 32 ATP. That's a much better option. However, it's really slow and it takes oxygen. However, glycolysis can just keep breaking apart those glucose. Okay, don't need any special factory or anything. We could just break it apart, make ATP, but we only make 2 ATP. Okay, and we have to put 2 ATP in. You get 4 ATP out, 2 net ATP. It's just not that much. Okay, So you can keep doing that, but this does not require oxygen, which means it's anaerobic. Aerobic respiration occurs over here. Anaerobic is over here. So your bacteria are going to do that, and it says it right there. Okay, we have both of them. This is the start for both, but if you don't have any oxygen, you're running low on oxygen, you're just going to be doing anaerobic respiration. Now, bacteria don't need oxygen to do this at all, okay? So bacteria, you can have them make any type of alcohol through alcoholic fermentation, because that fermentation process is going to break apart the NADH and give you NAD+. And when you have that NAD+, you can go back through glycolysis, and the ATP is going to get burned, don't worry. Every enzyme is going to be using that ATP. And so you'll get ADP, the uncharged battery, back. But you need that NAD+, the uncharged battery, back in order to start glycolysis. Of course, you need sugar, so you got to have sugar around. You know, bacteria need to eat sugar, okay? I need to be able to break down stuff for sugar, okay? Okay, so I think we're done with cell respiration. So the key with learning this stuff is to not let yourself get confused with the two, because they are similar. So you can use the knowledge that we learned in the last one to help with this, but you don't want to get confused with it, okay? So the next one that we're going to learn is photosynthesis, okay? In photosynthesis, we have a two-step, okay? The two-step reaction for photosynthesis is going to be the light reactions and then the dark reactions. My dark reactions here uses Rubisco, okay? And we are going to have, that's called the Calvin cycle, the light independent reactions. It could be called the dark reactions because, you know, it doesn't necessarily happen in the dark. Only some organisms or only some plants do that. But I've got this in green because we are thinking about plants. We are thinking about sun, water, and carbon dioxide coming into the plant to go through this process. So here's the zoomed out and here's the zoomed in, okay? So we have our zoomed out version here where we have water and light going into the thylakoids, and your thylakoids, the membrane of the thylakoid, okay, remember is like you get more surface area when you crinkle it up there, okay, and it can go out. And this is a zoomed in picture of the thylakoid membrane, okay. Now, there's a lot of things kind of happening here. Basically what you, at this level, we have photosystem 2 and photosystem 1. Photosystem 2 gets hit by light, and it rips apart water. That frees the hydrogen to be released into the thylakoid lumen. Your thylakoid lumen is going to be like represented, let me draw my thylakoid lumen. If this is our chloroplast, okay, then our thylakoid lumen, our thylakoid is going to be like this, and there will be an inside and an outside, okay? So inside, we've got here in blue where a bunch of hydrogen is going to be concentrated, and we know what happens when hydrogen gets concentrated, and it's going to be concentrated all inside of here, and when it's crowded on the inside, it's going to want to move out, okay? So when we see the hydrogen being concentrated in the thylakoid lumen, then we know that that's going to want to flow out. But it can't just flow anywhere through the phospholipid bilayer because these are polar molecules, and these polar molecules with the non-polar tails in the middle, they don't like charged things to go through them. Hydrogen, the proton, can't just go straight through there. It only goes through the ATP synthase, and which spins it, and that spinning, that motion, that is going to allow you to make a bond in an endergonic reaction, okay? Putting energy into a bond, adding a phosphate on to charge up ATP. Sounds familiar, but here it's happening powered by light, and it's happening in the thylakoid, okay? So our thylakoid here, this is the thylakoid membrane right here, thylakoid membrane. This right here is the outer membrane of the chloroplast, okay? And the space in between is the stroma. The stroma is where our second reaction is going to occur, and that's the dark reactions, which I was showing you here. The dark reactions is a cycle. Again, this looks similar to Krebs cycle. It's not. It is a cycle where you rearrange carbon molecules, okay? But the point of this cycle happening in the chloroplast is to make glucose. Here's a sucrose, okay? You know, here it says phosphoglycerate, okay? It's all just going to be made into glucose so we can make cellular structures. Cell walls are all made out of glucose. The structure of all plants, okay? It's just a whole bunch of glucose put together. We're just making this sugar and using it for, whatever we want, okay? So this thylakoid, this is like the kind of zoomed out version. This thylakoid membrane is where we're going to have our first reaction take place, which we have up here with photosystem 2 and photosystem 1, okay? And so I'm going to label that with our thylakoid membrane here. This is the first thing, and this will be called the light reactions or the light-dependent reactions because it depends on having light around. So these light-dependent reactions are going to, just to orient ourselves here, we've got photosystem 2, photosystem 1, and we have ATP synthase, okay? We have photosystem 2, photosystem 1, and ATP synthase. Photosystem 2, okay, right? So the reason we have it folded and we have a whole bunch of these all over in the chloroplast is because we have more surface area, so we can have more of these enzymes doing the work that we see here, okay? So when light hits photosystem 2, it activates it, and that energy allows us to rip apart water, exergonic. Okay, use the energy to rip apart the water. Releases that energy, and that energy bounces around, pumps some hydrogen in here with another protein we don't need to know about. And then the electron, which comes from the broken bond, the electron comes over here, and then, things happen, complicated. And then it goes over here. By that time, the electron has done a lot of work and needs to get charged up again. So when light hits photosystem 1, it's going to get charged up and really excited, and that electron is going to bounce around and allow us to charge up NADPH, which is a different protein, okay? Or a different energy molecule, okay? Now that battery, NADPH, gets charged up here by photosystem 1, okay? So photosystem 2 charges up ATP and rips apart H2O. Photosystem 1 charges up NADPH, okay? All right. Now that's cool. That's all well and good. We've got NADPH, okay? And then we've got, a bunch of hydrogens inside the thylakoid lumen. So they're concentrated, so they want to flow out. That's a concentration gradient, the rules, okay? And it can only flow through here. Then we got a spin of ATP synthase, and that is going to allow us to grab a third phosphate and slap it onto the ADP, an endergonic reaction, charging up ATP, okay? Now that's the first step. The first step here, the light reactions, okay? Light dependent in the chloroplast, right? Then the whole point of making those batteries is not so the cell can use it for anything, okay? The cell is specifically going to use it for step 2, which is the Calvin cycle, the dark reactions, the light independent reactions, which light can be shining and this can occur. It's not that light turns it off. It's just, it doesn't need light, okay? So we won't have any proteins getting activated by light here, okay? Now there's a whole bunch of enzymes that are doing this rearranging of these molecules, but the main one we need to know is rubisco. Rubisco is what's going to take the carbon dioxide from the air, which plants are taking in from the bottom of their leaves, open up a little stomata. Yep, plants breathe carbon dioxide, we know this. Rubisco is that enzyme which can take carbon dioxide and fix it to ribulose 1,5 biphosphate, if you want to know where the name comes from. Rubisco, okay? Ribulose, biphosphate, and carbon dioxide. That's the enzyme that puts it together, okay? Well named, it makes sense. And then we are able to rearrange. Here's the ATP that we're using from step 1. The light reactions gave us the ATP, which we are using. We're breaking off that third phosphate so that we can rearrange. Make a bond, break a bond, make a bond, break a bond, okay? Then here's the NADPH, which we need to use and pop out NAD+. Break that hydrogen off, use that energy for rearrangement. There's ATP being used again, okay? And why do we have so many different steps here, okay? Why do we have 2 different batteries and so many different steps? Because evolution doesn't do things the simplest or easiest way. Evolution just gets stuff done. Does this get it done? Yeah. Is the planet green because plants are using this? Yes, it works, okay? We don't, all right. So like I said, the light reactions, the whole point is to charge up the batteries so that you can then do the Calvin cycle, the dark reactions, in order to make our glucose, our sugar. And all we need to do then is bring in carbon dioxide for that Calvin cycle. That's going to use the charged batteries and spit the charged batteries back out to the thylakoid membrane, okay? Now, just to zoom out again, our step 1, light reactions, is occurring on the thylakoid membrane, and then those charged batteries are just spit out into the stroma, okay? So we'll see that the charged batteries of ATP and NADPH are just being spit out, okay? Spit out into the stroma. Out into the stroma here, we have all the ATP and NADPH, so that the Calvin cycle can just happen out here in the stroma. Step 2, Calvin cycle. Step 2, Calvin cycle. So it's happening all over the place. That's step 2 Calvin cycle, okay? To charge up glucose, okay? And that's what makes energy, all right? Now, of course, we need to make sure that we are comfortable with these graphs, making bonds and breaking bonds and changing the energy states, okay? We have this exergonic is called catabolic, like a catastrophe. And this anabolic, we are adding something on, okay? And this one, yeah, this is exergonic, this is endergonic. This is typically, okay, so here's the other thing that's really important to write so that we're comfortable with this. This breaking a bond is called hydrolysis, because the way you break a bond is you add water into it. Now, water did some things that we discussed before. That's not what I'm talking about here. Anytime you do want to break a bond, you will see water being added, okay? And that takes the place of the bond. Don't worry about that necessarily, okay? But the making a bond, which we have right here, that all of those processes, every enzyme in the body that is making a bond, it is dehydration synthesis. We are pulling out 2 hydrogens and an oxygen. If you pull out 2 hydrogens and an oxygen, you make water as a waste product, and in water's place, you can form a bond, okay? So when we say break a bond by cutting with water, hydrolysis, and we say make a bond, dehydration, we are literally taking out 2 hydrogens and an oxygen, or putting in 2 hydrogens and an oxygen in order to take the place of a bond, or to be pulled out so that a bond can be there in its place. But this takes a lot of energy to make that bond of a higher energy molecule, and this takes a little bit of energy to get over that activation energy to release that energy. Okay? I think that's pretty good, and it's shorter than the last one.